Just like you don't want smelly trash building up in your house, you don't want NADH building up in your cytoplasm. NADH is kind of like the waste product from catabolizing or breaking down molecules, except it's way better than normal waste because you can typically cache in the electrons from those NADH in order to make ATP. But that requires oxygen, and it requires that the NADH be in the mitochondria. If you got a bunch of NADH building up inside of your cytoplasm, well, that's actually going to inhibit glycolysis. So in order to keep glycolysis or sugar breakdown going, we have ways like fermentation to regenerate NAD+. Another compounding factor that is less appreciated is that the NADH that we generate in glycolysis can't actually get into the mitochondria where the caching in occurs. Therefore, the NADH that we make in glycolysis has to be kind of shuttled in, or the electrons from it have to be shuttled into the mitochondria. There are a couple of different shuttles that you can use for this, and they come with kind of different energy payouts. So let's talk a little bit more about how we can regenerate NAD+, and how we can get our cash in that trash. Let's start by thinking about what happens when your cells go to break down glucose, so blood sugar. The first step is that glucose is going to be broken up in the process of glycolysis. Glycolysis is going to generate a couple of NADHs and is going to generate pyruvate. Pyruvate, well, then that can get oxidatively decarboxylated to give you acetyl-CoA, and that acetyl-CoA can then enter the TCA. In the TCA, well, here you're going to generate even more NADH, as well as FADH2, and the electrons from these can then be used to power the pumping of protons into the intermembrane space, which can then power the synthesis of ATP. In order for this NADH and the electrons from it to be useful, you need to have them present in the mitochondrial matrix. Now, this is a problem because cytosolic NADH can't actually get into the matrix, and so later we'll discuss how we have to use shuttles to get reducing equivalents inside the matrix instead. But for the electrons that are generated in the TCA, the NADH is generated in here. Here you're in the right place, so all's good to go. However, the electrons that you are passing off to NAD+ in the process of glycolysis, well, these are actually going to be taking place in the cytoplasm, and you're then going to be building up your NADH, where what you want is your NAD+. So because NAD+ is typically going to be used for these catabolic or breakdown reactions, we need to keep it in its oxidized form. The power of its reduction power and things like this is going to be influenced in part by the relative concentrations of our NAD+ and our NADH. We want to keep, therefore, a ratio of NAD+ to NADH that's going to be very high, meaning we have a lot more NAD+ than NADH in our cytoplasm. In order to do this, we're going to have to keep regenerating that NAD+ if we keep kind of using it up during glycolysis. This is especially important because what happens is that the NADH is actually going to directly inhibit glycolysis, as well as the citric acid cycle. This is a key example of negative feedback. What happens is that if you're building up a lot of NADH, it's going to inhibit key enzymes in these pathways. Because it's kind of saying like, okay, slow down, we're building up down here. We've got enough of this. Start doing other things, start doing more anabolic things. So NADH, not only is it a product of the reaction, and so if you have NAD+ building up, that's kind of like you're building reactants, but if you have NADH, that's building up products. So that would be unfavorable because, well, you want reactants and not your products. But also that NADH is going to act as an allosteric negative inhibitor. So it's going to bind to these enzymes and prevent them from doing their job. And so This NADH is going to affect the pyruvate kinase as well as the pyruvate dehydrogenase complex. So you're going to be inhibiting your phosphoenol pyruvate to pyruvate conversion, as well as the actual decarboxylation of pyruvate to give you that acetyl-CoA that can injure the TCA. And within the TCA, you also have inhibition from this NADH. And so you need ways to regenerate the NAD plus in all these cases. If you're in the mitochondria and there's plenty of oxygen, well, then you're going to regenerate it through the electron transport chain. You pass those electrons off to complex 1, and this is actually going to help power the production of ATP in addition to regenerating the NAD plus. So all's good. It's still good to have this inhibitory mechanism, however, because if your electron transport chain can't keep up, if you're doing enough of this catabolism, we got plenty of fuel breakdown, let's go and store some or something like this, well, then you're going to want to inhibit further breakdown. And so it still makes sense that you would have the NADH be inhibiting in this case. But about glycolysis. So glycolysis, in order for it to keep going, it's also going to need to regenerate that NAD+. The NADH that is made in glycolysis, it can't directly enter the mitochondrial matrix. So how are we going to get rid of that excess NADH in your cytoplasm? Well, it depends. If we've got plenty of oxygen, well, now we're going to use one of these shuttles in order to get the electrons from NADH into the matrix. This is great because then we can actually use it to generate the production of ATP. There are two main shuttles that we can use for this, the glycerol 3-phosphate shuttle and the malate-aspartate shuttle, which we'll discuss in more detail in just a minute. But the other option is if you don't have oxygen, well, now you can go a different way. So remember that the electron transport chain, it depends on the presence of oxygen. And so if you don't have oxygen, you're not going to do the electron transport chain. So even if you were to shuttle those electrons into the mitochondria matrix, well, they're not going to be useful because you can't do that whole EDC. So instead, in these cases, what you can do is you can do fermentation. Some cells rely on fermentation, such as your red blood cells, because they don't actually have mitochondria. So your red blood cells, your erythrocytes, are always going to depend on this fermentation. But also, like your muscle cells, well, if they're working really hard, they're going to then have to go under, not going to have enough oxygen to keep up, and they're also going to go through lactic acid fermentation. So what is fermentation? Basically, fermentation is just where we regenerate NAD+ without oxygen. And this is going to allow glycolysis to continue anaerobically. So we do lactate fermentation, but other things like yeast, plants, and microbes, they can do alcohol fermentation. But in us, we're basically using lactate dehydrogenase, and we're generating lactate from pyruvate. That lactate can then go, and it can travel through the blood into the liver. And the liver, well, here you can do gluconeogenesis, so that liver can actually take that lactate, convert it back to pyruvate, and that pyruvate can then be used to generate the production of more glucose, which can be sent back to your muscle cells so that they can keep going. Sweet deal, right? In this case, the muscle cells were getting rid of that lactate, or they were getting rid of basically the NADH, but without actually shipping out the NADH because they still need the NAD+, so they're passing the electrons off onto pyruvate to get lactate. Now they've got this NAD+ that they can use to keep powering the production of ATP through glycolysis. But remember that glycolysis, well, here we're actually not going to get that big of a payout directly in terms of energy. In the process of glycolysis, we're only generating 2 ATP molecules. Contrast this with what we can get from oxidative phosphorylation, where we can get like 26 to 28 molecules. And so how are we going to get all of those extra ATPs? That's going to be happening because of those electrons from molecules like NADH and FADH2, and to power the pumping of protons, to power the ATP synthase to make ATP. So if we could get our NADH from glycolysis, well, now we'd have the opportunity to use it to make ATP. Fermentation just allowed us to keep going with glycolysis, but it doesn't allow us to get the cash-in payout from the NADH that we generated with glycolysis. And because we're kind of stopping at the partway point, we're not getting the energy that we would get from breaking down the pyruvate further. So fermentation is great if you don't have oxygen. But if you do have oxygen, well, now you're going to want to go and you're going to want to take these NADH and you're actually going to want to cash them in for energy. So let's talk about the shuttles that are going to allow us to do this. A quick note about shuttles. Basically, this is just a way where we can transport the equivalent of one molecule from one place to another. And what we're doing is we're actually kind of disguising the molecule. So we'll change its form back and forth. And so hopefully it'll make more sense in a second. But there are these two main shuttles, the glycerol 3-phosphate shuttle and the malate-aspartate shuttle. One of them is going to, so the malate-aspartate shuttle is going to let us come in at complex 1. And the glycerol 3-phosphate shuttle, we're only coming in at complex 3. In order to really understand this, let's kind of just review really quick the electron transport chain. Basically, you have this series of membrane-embedded complexes that are going to pass electrons from one place to another. So you have this complex 1, it's complex 3, and the complex 4, all of which are going to pass electrons from one thing to another, pump out protons, and then those protons that were pumped out, you get this gradient building up that's going to power the production of ATP. How it works is that complex 1, well, this is where you take the NADH, and this is where you're regenerating that NAD+. So if you have NADH in your mitochondrial matrix, the electrons from it can be passed off to complex 1, which can pass those electrons off to Q, which is a membrane embed, like a membrane molecule that can move through the membrane, and it can take those electrons to complex 3, and then complex 3 is going to pass those electrons from one thing to another, pump out protons, and pass the electrons on to cytochrome C, which is a little protein that can then take those electrons to complex 4. Complex 4 is going to pass off those electrons, pump out some protons, and ultimately you get this gradient of protons built up that can then flow back through like one of those water wheels and generate the synthesis of ATP. So what about this complex 2? Well, complex 2, this is actually the succinate dehydrogenase from our citric acid cycle. So it's this step right here. In this step, you're generating FADH2. The FADH2 is going to basically enter the pathway at this point, and it's going to pass those electrons onto Q. So that would be this. But there's actually a lot of different ways we can get electrons to Q. So we had one way was our complex 1. We had another way was our complex 2. And now let's talk about a third way, which is going to be our glycerol phosphate shuttle. So in this glycerol phosphate shuttle, what's going to be happening is that you are taking this, the NADH that you generate in glycolysis, as well as dihydroxyacetone phosphate, which is something that you could also generate in glycolysis. So it's this step. What you're going to do with it is basically you're going to transfer the electrons from NADH onto dihydroxyacetone phosphate. That is, you're going to reduce the dihydroxyacetone phosphate to form glycerol 3-phosphate. Now, glycerol 3-phosphate, we have this enzyme glycerol 3-phosphate dehydrogenase that's on the mitochondrial matrix. I'm sorry, the inner mitochondrial membrane, but in like this inner membrane space side. So we don't have to actually get this into the cells, into the matrix. But we can take this glycerol 3-phosphate that's hanging out here in this inner membrane space. And now what we could do is we could oxidize it back to dihydroxyacetone phosphate. In the process, we pass these electrons from FAD to FADH2, which can then be passed on to Q, which can then enter complex 3. So this is an alternative route to getting electrons to Q, and then we can go to complex 3. Importantly, we're passing the electrons from this NADH, we're generating NAD+, we don't actually have to get any molecules into the mitochondrial matrix, and it's easier to get into the intermembrane space than it is to get into the matrix. And so we can have these molecules all hanging out in this intermembrane space without having to worry about actually getting the molecules directly into the matrix. Because we're entering at complex 3 rather than complex 1, well, this means that we're not going to pump out all those protons that we would at complex 1. So instead, we're only going to pump out 6 protons. Now, it turns out that it costs about 4 protons per ATP made. It's about 3 protons per turn of the cycle. And then there's actually a 4th proton that is going to be needed in order to bring in another phosphate group. So we say that there's like 4 protons per ATP. And so if we had 6 protons being pumped out, well, now what's going to happen is that this glycerol 3-phosphate shuttle is going to make about 1.5 ATP equivalents per NADH that we had out in the cytosol. So that's the main way that your brain and muscles use to get these electrons into the matrix. But what about your other cells? Well, it turns out that your heart, liver, and kidneys, they're like, hey, we want to get in at complex 1. We can get a bigger yield there. And so they use this method called the Malate-aspartate shuttle. How this works is a bit more complicated, but don't worry, most of this is just to regenerate all the components that we need. Because in this case, we're actually transporting molecules through the membrane, not just transporting electrons. We're going to sneak the electrons from NADH into the matrix. So we're going to take oxaloacetate and we're going to reduce it to form malate. In the process, we're oxidizing our NADH to regenerate our NAD+ and get rid of that NADH. This malate then can enter the mitochondrial matrix where it can be converted back into oxaloacetate. And in the process, we're taking the electrons off of malate and putting them back on NAD+ to give us NADH. This NADH can then go and enter complex 1, go through the electron transport chain, and be used to make ATP. But if we want to keep doing this, we're going to have to regenerate this oxaloacetate. In order to regenerate the oxaloacetate, you might think, oh, why don't we just ship oxaloacetate out of the mitochondria? But oxaloacetate is another one of those molecules that can't get through the membrane. We keep a tight control over oxaloacetate concentrations, and it actually has like the lowest concentration of any TCA component in our mitochondria. It's tightly regulated. We can't ship out the oxaloacetate. Instead, what we're going to do is we're going to transform the oxaloacetate into aspartate, and then we're going to transport the aspartate out and then convert it back into oxaloacetate once we're in the cytoplasm. So it's a bit convoluted, but we're doing this because we need to regenerate this oxaloacetate out here that we transformed to malate and then shipped in. We have to take this long route because oxaloacetate can't get shipped out. So what we do is we take this aspartate aminotransferase, we take oxaloacetate, and we take glutamate. We're going to transfer the amino group from glutamate onto oxaloacetate, which is going to generate aspartate, and then we get this alpha-ketoglutarate. We now, if we want to keep this whole thing going, what we're going to need to do is we're going to need to take in more glutamate, and we're going to need to take out alpha-ketoglutarate. And so how we're going to do this is basically the glutamate aspartate antiporter is going to allow us to ship out that aspartate that we needed to regenerate the oxaloacetate and ship in the glutamate so that we can redo this reaction. This malate is also going to be kind of swapped for something. In this case, it's going to be swapped for alpha-ketoglutarate. So the alpha-ketoglutarate, that was kind of the waste from this aspartate aminotransferase step, is going to be a reactant in the cytoplasm when we need to use it to regenerate this oxaloacetate out here. because out here, you're basically doing the reverse reaction with the cytosolic aspartate aminotransferase. You're passing the amino group from aspartate onto alpha-ketoglutarate to give you glutamate, but more importantly, to give you the oxaloacetate, which then you can pass electrons through to get malate, ship the malate in, and take those electrons, pass them back to NAD plus and get NADH that's now in your matrix. So this is just involving some like transamination reactions and some molecular swapping. What's going to happen in this case is that because your NADH is in the matrix, it can go in at complex 1. Well, here you're going to get 4, 8, 10 protons pumped out. And we said, remember, that we needed about 4 protons per ATP. If we're generating 10 protons, well, now if we do 10 divided by 4, that's going to give us 2.5. So if we go in with the malate-aspartate shuttle, we're going to get about 2.5 ATP from that cytosolic NADH. And if we were to go through the glycerol 3-phosphate shuttle, we would only get about 1.5. That'd be equivalent of one of those FADH2s that we got from complex 2. Remember that this is just a different way to get electrons to Q. This is why, if you, at the end of the day, if you go to do some ATP accounting, you'll see that the final amount of yield per glucose is going to depend on a variety of things, but part of it is going to be whether that NADH was coming in using the complex 1 from the malate-aspartate shuttle, in which case you could get 5 ATP, or if it was coming in through the glycerol 3-phosphate shuttle, in which case you could only get 3 ATP. But those are just all estimates because, well, the production of ATP is actually dependent on the gradient and not the source of the gradient. So at the end of the day, you're regenerating that NAD+, you're getting rid of that NADH, you're therefore building more reactants so you can push things forward, and more importantly, you're taking away the NADH that would be inhibiting these regulatory steps. So keep going on the breakdown until we start building up. Once we start building up, let's inhibit things, let's go and let's build things or do whatever else we want to do.